Craig Venter once said, the possibility of life in the future depends not only on our understanding and use of DNA, but perhaps a creation of new forms of life forged not only by Darwinian evolution, but created by human intelligence. Maybe that day is here. Maybe that day is here because today we meet to discuss about SpudCells. That's right. Welcome back to the latest update series of Drishti IAS. This is Divya Kawatra, and we are talking on a very interesting topic today that is called SpudCell. What is SpudCell? Let's find out, and how it relates to creation of human life in synthetic forms. All right, let's find out. So to understand SpudCell, first of all, let's understand what is the current development that has happened. Scientists have created world's first man-made cell. That's right. It is world's first man-made cell. But wait, this has been done before. This is not a new discovery. So what is special about this? This is world's first man-made cell that can eat, grow, and reproduce. This is a cell that on itself, synthetically produced, can eat, can grow, and can reproduce. That is an amazing discovery. So this is called SpudCell. The students have interestingly called it Spud on the name of potato because of the shape that was given to it. It looked like a potato. So yes, they chose the name potato, and potato is called Spud. So this became SpudCell. All right. That is the name SpudCell. It is the work of Kate Adamala and her team of researchers in the University of Minnesota. Okay. Where has it been done? In the University of Minnesota. This gives scientists a better understanding of origin of life and can help create new medicine and solve some world's biggest biological problems. All right. A lot of problems. You'll understand the reproduction. You'll understand the creation of life, how life would have been created. We'll understand that from synthetic objects itself. Like there is a bubble of oil and water that is floating around, and it has a capacity to eat, it has a capacity to grow, it has a capacity to reproduce. Just think about it. It is changing the entire word structure and helping us understand the basic biology from a totally new perspective, right? And we will have new creations of life. Let's understand how this works, what SpudCell is and how it is working, all right? First of all, it is world's first completely synthetic. Please mark this, it is completely synthetic, because earlier when cells were created, what happened was scientists generally used some sort of value, some sort of element from a living cell, right? From a living cell, and from there they would grow an artificial cell. So the base actually was living. Here in this case, this is like from the bottom, from the very scratch of it, we have developed this cell from zero, from synthetic material using no living material. All right. Do you understand that importance? All right. This is a man-made cell-like system capable of entire life cycle. It lives a full life cycle. That is why it is quite significant. That means it can feed, grow, replace its DNA, and divide at the same time. This has been built from scratch. It was assembled completely bottom up from non-living chemical components in a lab. There is complete synthetic structure here, and nothing living, but no living bacteria has been used here, right? The name, it was engineered by Dr. Kate Adamala's team at University of Minnesota, and students have playfully given it the name SpudCell, right? She asked her students to give it a name. It looked like the potato, so they all gave it a name, SpudCell, and it became famous. Of course, it got accepted because it was very creative. And now let's understand how the cell works. Okay, how the cell works. So we have created a cell. We gave it a food bowl. Okay. We gave the cell a food bowl. It started merging and it created a nascent cell, and then the pinching cell, and then started dividing, regrowing, and finally creating multiple cells. This is the growth stage. This is the division stage. It gets divided, becomes two. There are two cells now. From one cell, there are two cells now. And then they go to G2, G4. Then G5 generation, that is the final evolved cell. That means it ate, it grew, it got divided, it started reproducing, right? It started reproducing. This is done. How is reproduction happening? In a natural cell, it happens in the skeleton form. Here there's no skeleton. Here there's no skeleton. And SpudCell pinches apart using surface forces. It pinches itself apart using surface forces rather than needing a skeletal force, right? Rather than needing a skeletal force. The genome blueprint here is that the instruction for life. The instruction for life, this is the smallest known blueprint, only 7 pieces are there, and this is the genome, the blueprint of the structure, okay, that has been prepared. Now, it doesn't just reproduce, it also evolves. It also evolves. How does it evolve? Understand, it gets specialized food collector proteins. This is the G5 variant. It keeps growing. It leads to G5 generation. And there at the G5 variant, it starts developing its own proteins to actually capture good food. You see this? This has been done on its own. We have not given it anything extra apart from the food. After taking the food, these are all the steps the cell undergoes. And after that, it starts developing specialized food collector proteins. The structure that you can see outside of the cell, right? This is food collector protein. And this helps us understand that these cells can move to survival of the fittest, right? Anybody who develops good proteins will be able to get good food and will be able to survive better, while in the natural cell, this doesn't work. Right? Exactly. That is our creation. All right, that is how it works. Let's understand in one more format, how it works. First of all, it eats. It takes resource acquisition. It is a liposome. That will be able to gather food. Okay, it has a tiny lipid bubble and it is programmed with a custom membrane protein. That membrane will attract the feeder bubbles. Okay, there are larger bubbles and then there are feeder bubbles. Okay, so this protein membrane will attract the smaller bubbles and start eating it. Once it does that, the molecular soup, they touch, they fuse and allow the SpudCell to swallow fresh lipid, sugar, enzyme, everything. Okay. Once it eats, it starts growing, it absorbs the raw materials from the feeder liposomes and the membrane starts expanding. Just like you blow air in a balloon, right? The balloon is very small when you look at it, but once you start blowing air into it, it starts growing, right? It starts expanding. Similarly, the cell is very small, but the moment it starts eating up the neighboring bubbles, it starts growing, right? It starts expanding. That is what is happening to its membrane. Then it copies its code. The genome is uniquely small 90 kilobase pair split across 7 parts. Now this genome, they start copying its code and that copied code is again used to create more and more versions of it. Reproduction, right? But do you see that we have DNA matchings, right? Genetic matchings with our parents. Yes. So they are also maintaining that. They have 7 separate DNA plasmids, right? In humans, there are 3 billion. But here there are 7 separate DNA plasmids. And they successfully replicate the DNA to pass down to future generation. They actually keep replicating it so that they can pass it down to the future generation. Just like in humans, we have genetic and DNA capabilities of our parents. Amazing, right? Amazing. And then they can reproduce without a skeleton. Natural cells, obviously, they use a complex internal scaffolding that we call cytoskeleton. We have one skeleton outside of the cells, and then there is one small skeleton inside every cell of human being as well. That skeleton works at combining these cells. All right. And now they don't need the skeleton. They don't need the outer or the inner skeleton, and they can just join it together. Proteins can just crowd together and start formulating. All right. Then it evolves. Scientists gave some cells some genetic tweak. Right, like we have genetic modifications these days, right? So scientists thought that let's try this. So they gave one of the cell a genetic tweak, right? One of the group of the cell that they had a genetic tweak, made a very slight change, and they started evolving according to that, right? That they let them absorb the food faster. Okay, what was the tweak? That now they will be able to absorb the food very fast. The absorption that they are doing up till now, the speed will be very fast. They adapted to it. And they crossed at least 5 generations to the previous cell. One cell without the gene tweak and the second cell with the gene tweak, the second cell with the gene tweak is 5 generations ahead. That's how fast it is eating. That's how fast it is doing rest of the functions as well, right? And this leads to the survival of the fittest. Like if you are fit to absorb more food, reproduce quick, then obviously you will survive more. Clear? This leads us to our natural selection of survival of the fittest. The fittest cells will survive, rest of them will subside. Clear? This is how it works. The 4 hallmarks of life: eating, growing, genetic or DNA transfer, and reproduction, right? All the steps are done, and along with that, it is evolving also. A special step that it is evolving also after genetic tweak. Clear? Yes? Do we understand that? Very nice. Why is this significant? It is proof of evolutionary selection, right? They've given genetic variation to some cells. There's efficient gathering, 5 generations faster growing. So that means there is evolutionary selection. Then tiny eco-factories. Because the genome is modular, obviously it can be programmed like software. We can program it like software. They can be hyper efficient, low carbon micro-factories. Why? They can grow eco-friendly plastics, medications. They can do all these kinds of works, right? Then we have the caveat. It is not fully independent yet. SpudCell is massive leap forward, but still it depends upon the laboratory, its environment, proper environment that it requires. So for now, it is not completely independent. It is independent of any living cell. It is independent of any living bacteria, yes, but it is not completely independent because it still depends upon laboratory and the environment, right? So that is still a requirement. It has an instruction to read DNA, but cannot manufacture its own ribosomes yet, right? It is the liposome, but it cannot manufacture its own ribosome yet. That is the cellular machine that builds proteins. It needs the kind of food that will help it build the protein membrane and then strengthen the protein membrane, but it cannot generate its ribosomes on its own. That means it should borrow them from E. coli bacteria provided in the soup. Okay, in the soup itself, we have to give E. coli bacteria, and that will be used to produce the ribosomes. Clear? Exactly. This is how the SpudCell works. This is about the SpudCell, and I hope you understood. This is world's first man-made cell that has been made completely from the scratch in a synthetic manner, made in the University of Minnesota, and it is called SpudCell. All right? Okay. So we have a question for you based on this and this information that we've just shared. All right. Just read the question. Give us your answer in the comment section. Okay. All right. That's about it. Thank you so much for joining in. See you in the next session. Till then, take care. Thank you. Bye-bye.
For more informative content, like, share and subscribe, and do not forget to press the bell icon to get the notifications.